Okay guys, since we didn't get through the activity yesterday, these are your drawings of what you should have seen. Not only is this video for you to fill out your Meiosis N5 drawing notes, but this is also for you as a reference. You can always come back to this video and you can study for your test that is on Friday. Okay, so we start out with interphase, and our starting DNA has 4 chromosomes per cell. Now, this chromosomes are not coiled up into chromosomes yet. They're just chromatin, which is why we just have the beads sitting in the nucleus. So there's 4 different colors. You have the blue, the green, the yellow, and the orange chromosome. Okay. Now, I'll tell you when you get to your first drawing, because we're going to go through interphase and prophase as stages. So interphase starts out, and you have normal life of the cell in G1. Then you get to G2 and the DNA is duplicated in the S phase. So this would be one that you draw. So if you would pause the video to give everybody a chance to draw this. Okay, you should have your DNA drawn as chromatin, which we know is just the squiggly lines, kind of like this. Okay, then we get to prophase. Now again, don't draw yet for prophase. I'll tell you when. Okay, first thing that happens is that duplicated DNA condenses into what we call chromosome, into that iconic X shape. Okay, with that X shape, for you guys to draw it, what I would do is I have that white dot in the middle of each one. That is my centromere, that connecting piece for each chromosome. Okay, and I would do that, and then each one has 3. So you have 1, 2, 3. Now, obviously, yours is going to be a little bit better because I'm using a stylus on a tablet. Okay, so that's how I would draw each chromosome. So you'll have a blue, a green, a yellow, and an orange one. Each one has 3 little beads on each side. Remember that these are sister chromatids. So you have your original over here, and then you have your copy over here. So in prophase 1, homologous chromosomes are going to find each other and they're going to pair up. So my blue and my green are going to be homologous chromosomes. Right there is going to be blue and green, and then my orange and my yellow are also going to be homologous chromosomes. Now, a key thing that happens in prophase 1 is that crossing over occurs. So if you look here on my yellow and my orange, you'll see that these inside legs right here have exchanged DNA to where they cross over. So they flip genetic information. Remember, homologous chromosomes have the same traits on them, but they might say different things. So if the trait for eye color was here, this one, or the orange one might say brown eyes, the yellow one might say green eyes. They exchange that DNA and it increases genetic variation, which means it allows you to have variations of your parents' genes instead of having the exact same ones. It only exchanges between homologous chromosomes. Remember, also in prophase, our nucleus starts to go away, so you get the dotted line nucleus, you get centrioles and spindle fibers. Go ahead and pause this and draw it. Okay, for the rest of these, I'm pretty sure you just draw this. I'm not going to break it down into stages. So whenever you hit one, you might want to pause and then listen to the explanation. For this one, you have metaphase 1, which is where homologous chromosomes are going to line up in the middle of the cell. Remember that M for metaphase, M for middle, and so homologous chromosomes are what line up in the middle. Then in anaphase 1, we're going to split those homologous chromosomes. So one goes to one side of the cell, the other goes to the other side of the cell. We get to telophase 1, and we get our nucleus back. The spindle fibers and the centrioles go away. Now, in this one, normally the DNA would go back to chromatin, but since you're just going to divide it again, that doesn't really happen. It's such a quick process, we're not going to throw it in there. And then in cytokinesis, you get your nucleus back. So we now have 2 cells. First division, you go from 1 cell to 2 cells. Now we get into our second division. So we have prophase 2. In prophase 2, for each one, the centrioles and spindle fibers come back. The nucleus is going to disappear. Again, we still have our chromosomes, and some of them have different genetic material than others. In metaphase 2, this is exactly like mitosis, where the sister chromatids are going to line up in the middle of the cell. Sister chromatids line up in the middle of the cell. And then in anaphase 2, we're going to split those sister chromatids. So half of the X comes this way, the other half goes the other way. And this happens for every single X. Telophase 2, they get to either poles of the cell, the centrioles and the spindle fibers go away, and we get our chromosomes, single chromosomes that are in each nucleus that is coming back. Finally, we end with cytokinesis 2, which is where the nucleus is all the way back, centrioles and spindle fibers are gone, and we have just our DNA. Now, if you notice, the DNA is half the amount of what we started with, and I'll go back and I'll show you that in just a second. Okay, with cytokinesis 2, we end up with 4 daughter cells, and they are genetically different from the original cell. These daughter cells are going to be your egg and your sperm cells. Now, to look at what we start versus what we finished. When we started here, we had 4 chromosomes. We had the blue, green, the orange, and the yellow. When we finish, we only have 2, and they are not the same. It's actually a combination of all 4 because of that crossing over factor that occurred. So we go from 4 chromosomes to 2 chromosomes. This would be a full set of DNA, so that would be diploid, and we break it down in half, and that's where you get the haploid. So for example, if you have a cell that starts out with 10 chromosomes, and it goes through meiosis, to figure out what you end with in each cell, you actually get 5. You divide it in half. So for us, our somatic cells start out at 46 chromosomes, and then when we go to make the sperm and the egg cells, we divide it in half to get 23. It goes from diploid to haploid. Another key thing is that we make 4 total cells with meiosis, whereas in mitosis, regular cell division, we only made 2 cells. And the key thing is that they are genetically different. Because of that crossing over factor, they are not the exact same as the parent. Okay, that is your meiosis overview. Make sure you get those notes filled in. That N5, you need to have the name of each phase, a drawing for each one, and a description for each one. You will keep those in your binders, and I will check them when I get back on Thursday. Next assignment is to work on that worksheet that the sub will give you. That worksheet is due on Thursday as well. So I highly suggest you guys use your class time wisely so that you can be productive and get ready to review when I get back on Thursday, because you have a test over this on Friday. This video will stay posted, so if you need to come back to it, you are more than welcome to. Have a good day.